fun working with Sonia over the last couple of years. We came to her with the goal of automating and miniaturizing our single-cell RNA-seq pipeline. So it's been really a lot of hard work on her part and on the part of folks in my lab, and I think now we're just seeing the interesting findings out of it. So basically this is walking you through kind of our experience with adapting our pipeline to an automated system. So our motivation in getting into single-cell transcriptome sequencing was for two main reasons. One is to understand cellular heterogeneity. We knew that this was present in our cultures. When we did immunocytochemistry, we saw that the cells were not uniform in their staining. Even by morphology, the cells are not uniform in the dish. The other thing that we wanted to look at on a more invisible level, but that we postulated was going on, is that when we were doing bulk cell analyses, that we were seeing correlations that may not hold true within a single cell, but held true in the population, and also that we might be missing interactions that occurred within the cell, but was not apparent in our bulk sequencing data. So I won't go through this in detail. We actually have a paper coming out in the Journal of the Association of Laboratory Automation, so JALA, that talks about some of these ideas in a little bit more detail. So just in broad terms, this is technically how we approach the problem. So on the left, I've listed the challenges of single-cell sequencing, and these include the low input of the material, which influences the reproducibility and the sensitivity of any assay that one is doing, and the need to study large numbers of cells to get a sense of the population. And for this, we have challenges in terms of scaling our experiments and the cost of the experiments when you want to study a lot of cells. And then our idea was potentially that automation and miniaturization could attack these challenges in the ways I've listed on the right. So namely, for automation, we would get away from having the problems with reproducibility with doing things manually. And then we would enable processing of larger samples, speaking to the scalability issue. And then for miniaturization, that we could, because the reaction sizes would be smaller, we could preserve the concentration of the elements in the reaction while putting in a lower absolute amount of input nucleic acid, and then also reducing the cost of the reagents. So to begin with, we wanted to do essentially a set of proof of principle experiments to answer these questions. So first, do libraries that are constructed in low reaction volumes with low inputs generate reproducible data? And then second, do these libraries also accurately reflect what the answers that you get with standard scale libraries? And then finally, can we introduce automation in such a way that we can scale the experiments to give us biologically meaningful results? So this is our overall pipeline here. So in these experiments that I'll describe, we perform cell dissociation, loading capture, washing staining, isolating imaging, and then doing the cDNA synthesis, so the lysing, reverse transcription amplification, all on the C1 system from Fluidigm. And then we moved into the new territory, which was how to do the actual library prep using the criteria that we discussed above. And for this, we used the mosquito HTS from TTP Labtech, using the Nextera XT chemistry. And then finally, the samples were pooled and then put onto a sequencer and analyzed. So that's all at the top. At the bottom is kind of the biological outline of the experiment. This was part of a larger experiment, which I'll explain in the last few slides. But basically here we're focusing on two stages of differentiation of pluripotent stem cells, what we are calling stage 1 and stage 2. For each of those stages, we selected 2 cells, and then we, after the C1 reaction, we divided the cDNA into different technical replicates to do the Nextera library prep and multiple technical replicates at different reaction volumes. And the reaction volumes are shown here, 2, 4, and 8 microliters. Of note, the manual library preparation that's recommended by Fluidigm for downstream library preparation from the C1 is performed at 10 microliters. So the 8 microliter reaction was close to that recommended manual reaction volume. So in terms of the C1, this is what it does, or actually the first step we do. We dissociate the cells into a single cell suspension. Then we load them onto the Fluidigm. We've stained the cells with calcium and ethidium homodimer, and then we visualize, at least this is what we do in my lab, we visualize each microchamber and we note how many cells are in the chamber and which ones are alive and dead, and we select only those chambers that have a single live cell for downstream library prep. In our hands, the average cDNA yield is about 0.4 nanograms per microliters and 15 microliters that you take off of the C1 chip. For the Nextera library prep, these are the systems that we used. So Sohaila can give you a lot more details, and I think there's an HV over at the booth that you can take a look at if you'd like. My lab is also open for people to take a look at if they're interested in seeing these things in action. So the HTS is shown on the left. It is the smaller volume handler. It handles 25 to 1,200 nanoliters. And the HV handles slightly larger volumes, 0.5 to 5 microliters. And as I go on, it'll become apparent why the two instruments are important. Both of them can be run at 8 or 16 channel settings. They use positive placement technology to handle the liquids. The tips are disposable, so we can avoid cross-contamination from sample to sample. Okay, and these can be integrated into full pipelines. In our lab, we physically put the plates on and off, but they can be integrated into other pipelines. So this is a little bit of a zoom in on the actual tips. So here you can see the tip is shown here. The shaft is there. It goes through the polyethylene barrel here. And again, those are the volume ranges, and those are the features that are important about it. The really nice thing for us is that we can handle low and high viscosity liquids with minimal changes in our settings. So, you know, if something is very, very high viscosity, we may change the speed of aspiration or dispense, but for the most part, we don't have to do a lot of fiddling around with different types of liquids. There is a dead volume, but it's quite small. The way we set up the reactions, we actually use the mixing feature to mix the reactions as we're setting them up. So, okay. So in the single cell library construction phase, what we did is we would normalize the samples so that all of them are at 0.1 nanograms per microliter. We had done some experiments upstream to determine that was the optimal input concentration. And we had all done that at 8 to 10 microliters. Then we scaled everything down linearly. So a 2 microliter reaction would have less input than the 8 by 4-fold. Here we used, as I said, the Nextera XT library prep kit and the mosquito liquid handler to set up reactions. So the first thing, of course, to address the reproducibility question was to look at technical variability. So if you can remember from a few slides back, we had 2 cells per stage. So cell A and cell B are from, quote, stage 1, and cell C and cell D are from stage 2. And so what we show here is the reproducibility within each volume, so 2, 4, and 8 microliters for the first 3 bars in each set. And then the purple bar at the right of each set is all of the different reaction volumes compared to each other. And so looking at the coefficient of variation, what you can see is that the variation between the cells is quite similar, but more importantly, when you compare across the reaction volumes, you don't really get an increase in the coefficient of variation. You can see there is one error bar that's a little bigger there on the cell D on the small reaction volume, and that is because there's a single reaction at 2 microliters that was at the edge of the plate, and it was an outlier. it ended up giving us a lot fewer reads in that library than the other libraries. So that would have been a tip-off on our read summary page right away, that would have been a library that either we would rerun on the sequencer potentially, or even rerun the whole library. Over on the right are just two different ways of looking at variability. What we've done here is across the x-axis in both graphs are the mean counts per transcript. So each dot is a transcript. And then the coefficient of variation in that transcript measurement. And so you can see that very, very low expressed transcripts tend to have a higher variability. And you see that pretty much for any technology that you're looking at gene expression. And that tails off as you get higher and higher counts per transcript. Again, on the lower right corner, what you can see is it's kind of, there's a lot more blur in that scatter plot until you take out that single outlier sample. That's the inset, and then it looks very similar to all of the other reaction volumes and cells and things like that. If you want to look in more detail, this will be sent to you. On the lower right corner, these are box and whisker plots of the same data, just to give you a more quantitative view. Again, this is all in our paper that's coming out, so you can see that as well. Our next question was, can we recognize biological variability? And so for this, we did principal component analysis and dendrogram for the same data. So we have color-coded the different cells, and then we've indicated the different volumes. But as you can see, the libraries from each cell cluster separately from each other. The variability within each cell is quite similar, except again, our single outlier shows up there on cell D over on the bottom right. And you can actually also see it in the PCA as well. Here, the PCA, there's two PCAs because the one on the left shows the first two principal components, the one on the right shows you the second and third principal components. But as you can see, it's all quite similar. The libraries from each cell cluster very well, and the different reaction volumes don't cluster even within a given cell. Another way that we look at the quality of a library is its complexity, so the number of genes you can look at. And so here what we're doing is looking at the number of detected transcripts in each library, and we're overlapping them. So for the first row, what you can see is the results for cell A, and then you have cell B, cell C, cell D, and then the different reaction volumes from 2, 4, and 8 microliters across the top. So in here, we've separated out the different reaction volumes, and we've summarized the overlap in the 4 different replicates at each volume for each cell as a % under each little cluster, each Venn diagram. And you can see across all the volumes and all the cells, they're roughly about 70 to 80, between 70 and 80% overlap. And if you want to look at the number of transcripts, the numbers are tiny, but again, this is all being published soon, but it's in the sort of 2,000 to 3,000 total transcripts. Here, what we're doing is we're looking across the reaction volume. So how reproducible is the data between 2, 4, and 8 microliters in terms of detected transcripts? And here, we're running around 80% or a little bit more between 2, 4, and 8 microliters. So that's pretty good. It's basically very similar to within a given volume. And we've done this in 2 ways. We've done it on the top, I'm showing the union of transcripts for each reaction volume, so basically taking everything within each Venn diagram. And then at the bottom, we're looking at the intersect. And then finally, we just did some basic heat map, differential expression analysis heat maps between the 4 cells. And so you can see that the behavior of all the libraries within each cell is very similar. So for example, at the top, you can see the transcripts in red right next to the letter A, which indicate the genes that are highly expressed specifically in cell A. And then the next group is cell B, cell C, cell D. Then you can look at the bottom. There are some transcripts that are in common between A and B, or between A and C, et cetera. So this is quite, quite a large percentage of the transcripts are differentially expressed in a clean way between the different cells. All right, so this was basically looking at the behavior of our system, our pipeline, whether we could trust the results from it. So after we did this, we then ran our biological experiment. So I'll just tell you sort of in rough terms globally what we did. So again, the C1 AutoPrep system was used for the single cell separation, cDNA synthesis, and amplification. Then we moved on to the mosquito HTS. We were able to transfer the cDNA from the C1 chip to a 384 well plate. We performed picogreen quantification and normalization. The normalization we did not do on the mosquito because the tips, all 8 or 16 tips go down and do the same volume, so it does not do cherry picking. So we did cherry picking ourselves. Picogreen quantification, normalization, as I said, and then the Nextera library construction was done on the HTS. We then moved to the HV system, which is the higher volume system, to do ampure bead purification. We then went back to the HTS to do picogreen quantification again, renormalized the libraries for pooling, and then sequenced. So at the end of the day, we ended up doing 384 libraries in parallel. They represented these 6 stages of differentiation, as well as fetal pancreas and adult islet samples. And we ran 48 libraries per group. Just to explain to you what this diagram means, we start on the left with pluripotent stem cells. These are stem cells that express OCT4 nanomine SOX2, as well as having a characteristic morphology of pluripotent stem cells. These cultures in our lab, we verify that they're pluripotent, doing in vitro and in vivo differentiation assays. So those are the undifferentiated cells. We then induce them to differentiate down this path towards beta islet precursors. So we do this in a staged way. We look at the expression of these different marker genes at each stage. So what we did is we did the differentiation at each stage. We took samples out, ran them on the C1, froze the cDNA, and then once everything was done, we put everything together into a library prep experiment and ran all the 384 libraries at once. Just to give you some early data that we have analyzing these samples, so one thing that we do is that we do a lot of quality control on the data coming out of the machine. So after mapping, we actually clean the data and then we downsample everything to the same number of reads per cell. This minimizes the problems that we might have with poor sampling of transcripts, especially low expressed transcripts in cells that don't have a lot of reads. So we do throw away some libraries at the data analysis stage that don't have sufficient numbers of counts. But here, what you can see, it looks like a PCA plot. It's actually quite similar in concept. It's using an algorithm called non-negative matrix factorization. But you can think of it like PCA. So the first axis explains the most of the variability in the data, and the second axis the next most. And here what you can see is color-coded the different cells in our data set. And you can see that there's a progression from less differentiated in the lower right corner up through the most differentiated in the upper left corner. You can also see that there is heterogeneity at each stage of differentiation. And also that there's overlap from stage to stage. So this is, as you might imagine, quite challenging for computational analysis. And so we're in the process of applying a number of different types of algorithms to try to sort out what the important genes are, what the subtypes of cells are, and things like that. Just another visualization. This is gets kind of ugly with lots and lots of samples. But one thing to point out is that after cleaning the data and getting rid of the cells that do not have sufficient numbers of reads, we end up with, so this is an unsupervised clustering, you end up with some interesting insights into the experiment. So what we did is, again, this is unsupervised using all of the data. So the only thing we did to filter the transcripts was to demand that the transcripts in this data set have at least 5 uniquely mapped reads, sorry, not 5, 10 uniquely mapped reads in at least one cell. And so what you can see here is that you have good clustering by stage of differentiation, but you do have some intermediate clusters. So, for example, off on the very left, there's a cluster that is all adult islet, but the next one over has adult islet and fetal pancreas cells in it. You also see as you keep on moving right, it's actually becoming less and less differentiated in this particular graph, that there are mixtures in the populations of stage 4 and stage 5. Stage 3 is pretty clean. There's an undifferentiated stage next, and then stage 1 and stage 2, there are 2 clusters, and stage 1 and 2 cells sort of interdigitate. And that is very consistent with this NMF visualization, where you can see stage 2 and stage 3 are the purple and the magenta. And so you can see there is quite a lot of overlap in those 2 populations. So as I said, we're doing a lot of work to still sort out exactly what's going on with the signaling pathways in these different populations. All right, so I'd like to give credit to those in my group that have been working on this. Sergio here, over on the right here, he's a former postdoc who did all of the cell culture work. Robert Mori and Minu Srinivasan did the genomics part, so making of the libraries, as well as Sergio. Actually, Sergio was very key in doing those experiments as well. And then Kwang is my computational biologist in the lab and was responsible for much of the data analysis. So I can take questions if you have any. Thanks.